Okay, well, thank you very much for that introduction. And I, like Ina, have to apologize that I'm going to, right after I'm done and take your questions, have to leave for the airport. So I also apologize to my fellow speakers, but they canceled the flight that I had. I'd actually prepared this quite nicely so I could stay till the end because I'm getting a lot out of this conference. It's my first conference of this nature. I'm certainly learning a lot. I don't know that I'm going to teach you anything about Viz. But what I hope is to focus a little bit on process, even though I'll show a little bit of what we've been doing in this project. For me, it's really been a journey of learning about process in how to essentially build resources that engage a community of mostly bench biologists. And I think one of the most important things I learned halfway through that process is to hire some people that came from an HCI background. And it really transformed the way we approach the problems. And it really transformed the developer team that we have in the project and the bioinformaticians. So it's really some of the process things are probably more exciting to you than the visualizations. So just to give you a very high-level view, these are large-scale projects funded by the NIH. They're called Bioinformatics Resource Centers. And their objective really is to bring the totality of the genomics data, genomics meaning all omics-style data, to the infectious disease research community, which again is mostly composed of bench-style biologists, epidemiologists, people with no background in Viz, and mostly not using omics-scale data sets. And so some of the key features is that there are standardized annotations, and there's a genome annotation service. There's also an integration of data and tools, personal workspace with, basically if you register, which is free. Comparative analyses, which is the main subject of this kind of a talk and session, and of course the capability to download everything. So before I forget, this is a very large effort of a lot of people, and the ones in the picture here are the ones at the Virginia Bioinformatics Institute, but there's also a very significant level of effort from the folks at Argonne National Lab and the University of Chicago. So this is the use case, okay? And I'm very grateful to Carol Goebel, one of my collaborators in Manchester, to point this out. If you're one of these folks, right, which are the folks we are explicitly wanting to connect with those folks. This is what they're trying to do every day. There's all this really cool big stuff over here, but really I'm just trying to do my research. And if you think about it, what is everybody doing? They're generating data. They're trying to compare it to all this data. They're trying to derive results so that they can put them in their grants and papers. That's the use case. And we always come back to that as we think of this resource. There are a number of different, this is one way to slice and dice the resource. There's a number of different, if you want to call them data types. They're not really clearly always the same thing, and they're not always that well defined. And here's kind of a grid of how we think about it. Do we have a backend for this data type? Is there enough really data quantity to do anything useful? For a while in many of these areas, there just weren't. Do we have viewers? Do we have analysis tools? Have we implemented the bring your own data, the My Data Space? And have we built de novo pipelines for reanalysis and integration? Now, I'm not saying that by presenting it this way that our intent is to turn everything green. This is more of a status report. In some cases, we don't see a need to go further. In some cases, we don't see a need to go further, and then we change our minds. But that's kind of the way we approach thinking about it. This is the front page. I'm not going to go into a lot of detail about how to use the resource. I don't think that's the objective here. But just to say, from a sort of HCI perspective, one of the things we found, initially our front page was much more like a big table of stuff, right? Here's click here, get genomes, click here. What we found is people really liked action things, finding, identifying, comparing, browsing. If we presented it that way, we could observe through the analytics that the usage went up significantly. So that's one of the first things is people want simple action type things. We also didn't have a comprehensive search at the beginning, and we got really nailed on that after a while because a lot of people have grown up, especially the bench biologists, thinking that everything needs to look like Google search. And so if they don't have that, they're kind of confused about what to do, and if they go away, they tend not to come back. And then here is for people who actually want to spend a little bit more time than just hurrying up and hitting something and try to learn a little bit more about what they can do. So these are just some examples, right? We integrate genomics data, but most importantly, and this has been a big focus, and it's an ongoing focus, metadata. More and more we see this as a metadata-driven information system. Because by the time you have thousands of genomes, why would you be interested in number 553? And there's no way to become interested in 553 unless there are metadata tags. So we've now spent a lot of time working on that aspect of it and connecting metadata to just about everything we can. Then here, this would be cogent to spend more time on today, but I'm not going to. But we do compute trees, but we compute trees using aligned sets of all the protein coding sequences. And so that's a slightly different way of computing trees than, for example, 16S, which doesn't give enough resolution for the kinds of questions that a lot of the people are interested in infectious disease research. We also have a variety of other omics data types. I actually don't like the term omics, but every time I try to get away from it, the rest of the biology community tells me not to do that. So you can have these other types of views, which I think you've seen things like this before. We tend to leverage externally built tools. We're not in the business of building another viewer for something that already exists. But we also, one of the critical things we do is to connect them so that when you do something here, and if this is another view of the same thing, and both of them are available on the screen, that they're talking to each other. And we find that to be something that the biologists really care about. And then there are other data types, and I think at least one of the talks today is going to address these in more detail, but what we find is points of entry for epidemiologists tend to be more like stuff like this. Points of entry for people who care about diseases tend to be more stuff like this. And points of entry who people are really literature heavy tend to be more stuff like this. So what we want to do is make sure there are points of entry for the different styles of end users that we're interested in capturing. So this is really important. I'm only going to make a very simple and obvious point here, example in my point, but we have different styles of users. And I would say that for all computational scientists that we run into, it's trivial how to fix their problem. That's their interface. And anything else we put in front of them, they say, why are you giggling? How do I get all of that to go away and just get to this? But so their problem was solved from day one in the project. That it's not complicated for us. And by the way, one of the greatest uses of the project is download all the data by bioinformaticians and computational scientists. We don't want to throw that out because they get standardized data sets, but they just want, give me a place where I can go, I download it all, and I'm going to go away and do my stuff with it. And that is one of the biggest use cases that's supported in our project. And we don't want to lose it. But a minimal interface for most of the bench biologists, which are the vast majority of the community that we want to reach in terms of numbers, is stuff like this. And of course, you can imagine that the time it takes to deliver this and this kind of stuff, and the process by which you have to interact to get to these versus this is completely different. So how do we get there? Well, this is another main point. The way we have found to get there is to go and embed ourselves in the research communities that we want to achieve and do their research with them. And when we sit down and observe what they're doing, we can figure out all sorts of clever ways, because we know the technology, to essentially implement something that supports what they're trying to do. Now, that doesn't mean that all we do is essentially listen to what they're doing and say, oh, we're going to make it exactly that way. I mean, I think Steve Jobs is famously attributed to saying that nobody asked him for an iPhone. So you need to be very careful. The point isn't to just go there and essentially mimic them. You also want to suck them into a new realm of what's possible. But all of these came from essentially sitting with people, collaborators, and doing collaborative research that yielded papers, insights, and on our side, this was a way to get requirements. Because what we found is if we walk around and ask people, hey, what are your requirements? They're like, what are you talking about? And why are you bothering me? I'm trying to write my grant. I'm trying to get results. Come on. It's a tax to ask people for requirements. It's a benefit to them to sit and do their research with them. So it shifts completely the dynamic, but you're still gathering requirements. You just don't talk about it. So this is what, on the first case, this is what people were doing. We would sit with people, and this is an Excel spreadsheet, in case you can't tell, the famous Excel spreadsheet. And people were sitting there and essentially downloading data sets from NCBI and other places, organizing them painstakingly, spending hours and putting them in order, and then they learned, oh, you can color code things in Excel, and they were color coding them, and this is going on, and in 100 different labs, 100 graduate students are doing it over and over again. So one day we said, what if we gave you something like that, right? Just as this was just a working prototype that we actually prototyped in Excel. And he said, that's cool. And what if it filled in automatically based on what you, and they said, that's even more cool. So this is the way we implement it. It's just a heat map. But the point was that it came, we already knew heat maps existed, but it came from sitting and observing and noticing that what they're trying to do is build a heat map, and they're doing it in Excel. If we made it available on the website and they could essentially create the groups and essentially populate it by creating the groups and get it, would that make their life easier? And boy, they love that. This has been one of the most successful tools, and we can measure this not just by blah, 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 but by watching the analytics. But then we said, well, oh, that's cool. So that's cool. And this was originally because they wanted to see protein families across lots of genomes. So over here, you select your genomes. You can decide if they need to be there, if they don't need to be there. If it's optional, you can do all sorts of things. These different intensities talk about copy number. And then we said, well, you know, what else can we shove in there? Well, we can shove protein families as well. And so once we had that tool, we started to think about all the different ways in which it could be used without people having to readjust to a new way of thinking. So basically, this is what we did in this example, right? Before, they were manually transferring to Excel on a per genome basis their genomes from NCBI. They were then directly linking to KEGG pathway information. And then they were doing across clade analyses. You know, I want to look across a clade of organisms that I care about and see what's present, what's absent. And they were mapping that onto KEGG diagrams. So then we said, well, that's cool. We can essentially use this and put them all in one place and have the table view all here. And these are now talking to each other so that if I select things here, they get selected over there and so on. So now there's no more of that happening, right? They're just sitting in the resource doing it. And so, and we can argue about KEGG. Everything that I heard before here about KEGG is true, but what is also true is they've built a really nice system in terms of you just being able to grab the interface and use it. Now, you can clean up and have better data under it if you want, but the system has its strengths. And in the first 4 years of the project, we used the Pathway Tools Viewer, which for those of you who have seen it, doesn't look like biochemical pathways to a biologist, so they really hated it. We had lots of good reasons to want to be there, but we said, you know, if the end users, it doesn't fit their cognitive style, doesn't look like a pathway, doesn't help them think about their problem, we have to get rid of it. And it's painful to get rid of things once you've implemented it, but we did. The next big thing was everybody would go there and make these groups and do cool things, and then, you know, they get interrupted, and they shift the computer, go home for the night, whatever, and then they come back the next day and they got to build the groups and everything all over again, right? And it's not so good. So we had to do a lot of pain on the back end, but we essentially said, what they're actually saying is they want a persistent session where they can group things. Now, if we're going to do that, then for the system to remember, we have to ask them to register, which before we didn't. Registration is free, doesn't take any personal information, just needs to know your email address. But now you can register, and once you register, now everything that you want to group and keep and deal with is stored from one session to another. And we're in the process of also making it so that you can friend people so that they can see your things too. So if you're in a collaboration, that allows you to essentially do collaborative work. So you can see all public bacterial genomes and other omics data that we have, or just your group of interest. So now what happens is it turns out that, for example, if you're a mycobacterium expert, maybe you mostly just want to live in mycobacteria. I'm not saying you should, or it's a good or a bad idea, but maybe that's what you want to do. So instead of going all the time to all of Patrick, you just create all the sets of things that you want to work with, and you just go to that, and the rest of it you ignore. And so we then found out that people were very interested in starting to compare not only genomes, which was pretty well put in, but they wanted to look at the transcriptional profiles. And then we said, well, but there are already geo. And they said, yeah, but have you ever gone to geo and tried to compare anything? And blah, blah, blah. And many of you who have gone through this know this. And so this is another process thing that I want to share. So we got all this metadata structured. We cleaned up some things. And this is what's currently available. We just started this year to release this, and we're going to keep going on a prioritized organism list from NIAID. Whoops. But the critical thing I want to share, which some of you might not be able to see because it's down low here, is this essentially was a collaboration with some bioinformaticians that had developed the methodologies to do this in a mostly automated way. And I think that if you have big resources like this, and we've been pushing really hard with the NIH to do this, is to create a way for smart bioinformaticians, Viz people, and others to have a way to essentially work to share their work into a system that already has data and already has a user community. And this has really been a challenge. We still haven't convinced them on the right model that they like. But we've been thinking about things like prizes. We've been thinking about things like small grants, all sorts of things. And we prototyped the idea with these folks to show a result. And so this got stood up in much less time than it ever would have been possible to do if they didn't already have something that didn't fit perfectly from an implementation perspective because it was never designed to work with our system, but essentially was, from our perspective, a working prototype. And so as a result of that, of course, that stuff fits into here as well. So we didn't have to make a new viewer. You can bury down. And what started to happen is people who are interested in all those unknowns, people like Patricia Babbitt, who's already been mentioned, now all of a sudden you could start to look at how often unknowns were showing up in these arrays, and also, importantly, what they were showing up with. Okay, so this might start to give some ideas of what they may be functionally related to. And that was something that people really liked, this correlated genes, especially because of the unknowns. And oh, by the way, bring your own data was enabled. And again, that was a huge spurt because the moment people found out I can bring my microarray data set and compare it to all the other sets that are there and do these kinds of things, I'm in. Because people are trying to analyze their data and publish the results. The metadata, again, has been a huge, huge success story. I have to say that we are officially not allowed to do anything manually curated because in principle, bioinformatics has solved all the curation problems and everything is automated. However, we stole a word from Google and we said, yeah, but we got to wrangle the data. So we do data wrangling. We don't do curation because we're not allowed to. And because of my interests and background, but also where the whole community is going on infectious diseases, is this issue of host pathogen. This is problematic because the way the funding agencies are organized is there's a division of microbiology, and that's the pathogen side. And then there's a division of allergy, immunology, and transplantation, and that's the host side. And of course, like all divisions, you know, that creates walls, and walls are a problem. And if you want to work on both, how do you get funded to work on both? So it's not trivial, but we're just pushing ahead anyway. So once again, we started off with things that were, at least there was data, and we could do something, which is host pathogen protein-protein interactions, initially just human bacteria. And there are great resources out there. The question would be, how do you aggregate this data, clean it up, and then present it? Presentation, of course, we're reusing tools that are out there. Phase 2 is getting ready to be done at the Pathogen Portal. We're going to expand from just human to all of these eukaryotes. We're also going to include vectors because these are important hosts, if you want to think of it that way, of many of these pathogens. And there's also going to be additional eukaryotes, which are pathogens, and then these guys, which are whatever you want to think about them as. And now this is the number, and it comes through all of that. And here's the prototype right now. So there's this notion of how do you show, you know, which organisms are involved. You can show the protein-protein interaction, taxon-taxon interactions, and once again, we're leveraging this idea. You know, normally these aren't all showing up necessarily to clutter, but you can mouse over and do things like that. and then the table down here, because often the biologists really want to get to the table with the reference and the literature that supports this pretty picture. And so this is, again, the prototype. If you mouse over here, you'll be able to tell what kind of interaction is supporting that. These will be really interesting to look at dual types of infections where you have hosts infected with, say, a virus plus a bacterium, and also comparing interactions that vectors might have with bacteria and human hosts might have with bacteria. And the taxon view is going to look something like this. And again, this is once again, showing the same kind of keys and showing certain interactions. And we're going to now take that, because some of our end users have said, well, why don't you show the bacterial protein-protein interactions? Says, well, we weren't interested in that, but if you're interested in that, we can do that. That's going to make a lot more data have to come through. There's this other view, is what we call a disease view, and this is when we started talking to the people in the clinic, more like the clinical research scientists. They were much more interested in this kind of point of entry. They said, I don't think about a pathogen the way you think about a pathogen. I think about diseases. So we said, well, gee, how can we create a view that includes disease? And so then once again, we're leveraging mesh terms and things like that to create these. But basically, you have gram-negative bacterial infections, and rickettsia is an example. And here are some of the ones that are caused by rickettsias. And then on the other side here, you essentially have the pathogen, and this is just, once again, leveraging the kinds of resources that are available out there to show it. So there's this thing called the portal, which talks to all of the BRCs, and this is, for those of you who care about back-end plumbing, this is an interesting thing. And for those of you who care about broad collaborations across multiple large programmatic infrastructures, it's also sociologically very interesting. But basically, this is supposed to do things across all BRCs. We view it as the disease triangle, which if you ever did undergraduate plant pathology, you'd have heard of. Medical guys usually haven't. There's general program information. There's data that we're supposed to keep inheriting from all these programs that NIAID funds, and then says, oh, they're about to end. You got to go over there and grab everything and put it there, and so on. And then there's host response data. And in some cases, like I'm going to show you, the idea is rather than each BRC develop the same analytical capability, why not develop it once and have it serve all the BRCs? So the primary example of that last point is an RNAseek analysis pipeline, which all BRCs were saying their users needed. So sort of the equivalent of a genome annotation service, so it's software as a service, but for RNAseek analysis. And I have to tell you, our BRC, and I know others, we're already at the point where people were calling us up all the time saying, can I ship you a bunch of hard drives that I just got from my local sequencing core, and I have no idea what to do with them? And in the beginning, we said, sure. And then pretty soon we said, no, we have to do something about this. So we essentially released something at the very beginning of 2012. It's now got over 200 registered users, and it is, of course, having a lot of gigabytes and terabytes of I/O. There's over 3 or 4 terabytes of data already sitting over there. We don't want to become SRA. Clearly, we're going to become now under pressure to figure out what are our data persistence policies. We're going to probably have to move to a model where people are going to only be able to keep the data there for a while, and then it's going to expire. And this is, once again, because the majority of our community really is bench biologists. They're informatics naive. What they really wanted is, teach me how to do this. So sure, I'll upload the data, or better, we give a way for their sequencing facility to upload the data into the My Data space, and then it's revealed to them, but they still needed some assistance in trying to learn how to use this. And so, this is the assistance, the visualizations that support the analysis. And really cool is the idea that, okay, once they've done it, if they're coming with a bacterial, one of the key visualizations they wanted was this. So the portal transfers the results in a standardized way to the BRC. But if you talk to David Roos and his community, the UPathDB folks wanted to render it in the genome browser. It's the same file format that's going out. The rendering is left up to each individual BRC working with their community to figure out how they want to display it. So this is a shared resource that then feeds the results into the different BRCs for display. So I wanted to summarize. I hope that these are points that have stuck with you. I believe, after many years of doing this, that the real way to understand requirements is to do collaborative research. Yes, we're supposed to be a community resource and a database and not do any curation and so on and so forth, but the reality is, if we want to succeed at what we're mission-oriented to do, and we're not part of the research community in an active way, it's very hard to have any clue about how to get it right. So we constantly embed folks in groups. We also bring folks over, if they want, with their data to analyze their data to figure out what we need to build in the future. This is much better than asking them what they want, in my experience. And again, I want to say, we're not user-driven, we're user-centered. Because if you're just user-driven, you'll never invent the iPhone. Biological theme-oriented integrated data repositories, such as the BRCs, in my opinion, provide a unique opportunity to tackle Viz with lots of data, a user community, and a plug-and-play architecture. We're not yet at the point of having an app store, but we're certainly moving in that direction. And we would like to essentially engage all the smart people in the audience that want to essentially plug and play a Viz tool into our architecture. You can't do that today. If you have something today, let us know, email me. We'll work with you to figure it out. But the more examples we have of that, the clearer it will be to us on what would be the right way to implement that. And then providing analytical resources that allow uploading and analysis of my data, because that's the use case. It's all about my data. Give unique insights as to what they're trying to do and a huge incentive for them to engage. They're not going there anymore for any other reason than, oh my God, I can go do my analysis there. That's cool. And one of the things we've noticed is there are dozens of thousands of bacterial genomes that have never made it into GenBank. I think only a small fraction of that is because somebody doesn't want to share. The problem is it takes 2 weeks to submit a bacterial genome to GenBank. And it takes a couple hours and almost no money to generate it. Now, when genomes took $2 million and 2 years to sequence, if it took two weeks to submit it, not a problem. But like one of my collaborators said the other day, he says, I've just sequenced 100 genomes in my tiny lab. And I'm going to tell my grad student to spend 100 times 2 weeks submitting them? Not really. So basically, by uploading the data to do analysis, the data's already in the system. So the moment you're happy, you just release it, and it's public. So you've gotten rid of the submission problem. And of course, we've been telling GenBank forever, because we would like every genome to end up in GenBank. We'll give you an API to suck our data anytime you want. And of course, they can FTP it anytime they want. So we already have a lot, and it's going to grow even more, of genomes that are not in GenBank, not because we want that, simply because people are needing to get their results and they're working in the system. So that's all I had. Thank you very much.